skull morphology of sharks really influences their ecology and evolution, and looking at that diversity of it as well. And my advisor, Dr. Kenshu Shimada, and then my two committee members are Dr. Winsor Aguirre and Dr. Eric Nordstrom. All right, so I just want to start off by mentioning that there are noticeable changes that are being observed in species distribution as well as community structure, marine taxa globally in our oceans. A lot of different types of marine animals are forced to change their lifestyle strategies in response to the increased threat of things like climate change with rising sea levels, as well as the encroachment of human activity on marine habitats. Now, sharks are especially vulnerable. This is because the increased sea temperatures influence them, arguably even more so than some other marine species, and it lessens the effectiveness of their sensory systems, as well as compromising the efficiency of their sensory systems within their habitats and makes them more vulnerable. Before we dive too deep though, what are sharks, right? These are animals that belong to the class Chondrichthyes, or cartilaginous fishes, and it's divided into two subclasses, being the elasmobranchs, which have sharks as well as rays, and then the holocephalans, which are rat fishes or chimeras. Now, all these groups, they have an important array of sensory systems, and the ones that I'm going to be talking about today are electroreception, smelling or olfaction, seeing or sight, and then vestibular balance and the equivalent of hearing. And yeah, those will be the focus of the talk for today. And these sensory systems are really critical for sharks to survive in their environments, and things like changing climate and other factors in the oceans are really kind of forcing the utility of these systems to be adapted in some way. So overall, sharks make up 9 living or extant orders, 37 families, 106 genera, and then over 536 species. I'm going to be looking at sharks from the genus context, so from the genera today. Now, some sharks, or some genera are very speciose. You might see the Carcharrhinoformes, which has over 40 genera, and some are smaller. So there's Echinorhinoformes, for instance, and Squatinoformes, and Heterodontiformes. They all have one genus. Another important thing to talk about here are the habitat forms that sharks occupy. So I separated into 3 main forms, that being benthic, which just means bottom-dwelling sharks. And then I have the demersal forms as defined as sharks that are swimming just above the ocean floor. And then pelagic, which is free swimming within the water column. And I define these classifications based on Eber et al.'s 2021 descriptions in their book, Sharks of the World, A Complete Guide. Oh yeah, and you'll see that there are bolded letters here. Those just correspond with the photos above. So another important aspect of their lifestyles is their feeding forms or their diets. I used Eber et al., their descriptions, again, to define 3 major feeding groups, which would be the suspensivorous. Sharks are those that consume microscopic prey like zooplankton and krill, so basically filter feeders. Macrophagous forms are those that consume kind of a wide variety of macroscopic prey, such as things like small marine fishes or larger marine mammals. And then durophagous forms are those that eat hard-bodied or hard-shelled organisms, such as crustaceans or mollusks, echinoderms. And again, I have these tables separating them with the bold denoting each of those taxa in the pictures. All right. So there were also body forms. And body forms, these are based on Stearns and Shimada's body forms that I'm going to be looking at as well. Basically found out that there were 2 main body/swimming body forms in sharks. One of them being the group A sharks, which if you think about that, they were referred to as shallow-bodied, and shallow-bodied basically means more like flat, so think of like a dinner plate. And then group B sharks were deeper bodied, so think of more like a round salad bowl as that analogy. I also want to highlight that, I remember I'm talking about the neurocranium today, and this is just an overall skeleton of a typical shark that you'll see, and this is kind of what we're going to be focusing on today, so I'm going to sort of zoom in to see that neurocranium there. Now, sharks are considered keystone predators. Now, what this means is that they are responsible for regulating populations of other marine animals as well as their habitats, just for the overall health of their environments. And it's the four senses that I previously mentioned, electroreception, smelling, sight, and vestibular balance, that effectively lets these sharks function as these keystone predators. Now, these senses are exhibited by organs that are situated in the neurocranium. So there's specific neurocranial regions for these. And I define these as the rostral region that houses the electroreceptory organs, the olfactory region that houses the smelling organs, the optic region that houses the eyeball for sight, and then the otic region here that is responsible for vestibular balance. And I just want to highlight here, before I highlight, in brevity, these are going to be called RU, so rostral optic otic, and rostral olfactory otic, to be called RU for the rest of the presentation today. I want to highlight also that this semicircular canal, which is stained in red, you can see that clearly here, but in typical skulls, you're not going to be able to see that because they're actually internally visible only. And so the otic region is really, I'm approximating the length of that semicircular canal for that. So the shark's skull or neurocranium has been studied for a long time because it has morphological simplicity that can be used as a sort of template for comparative studies of cranial anatomy across different groups of vertebrates. And just to give you some examples, by looking at neurocranial morphology, previous studies have been able to give more insight on shark feeding mechanisms. They've also been able to determine important aspects about their evolutionary history as well as their sensory systems. And these studies have definitely contributed to our understanding of shark neurocranial morphology, but they usually focused on like one specific group of sharks or maybe some genera. And this is kind of where my questions come in. So they've definitely shown that neurocranial morphology does have an influence to some degree on lifestyle, but I want to see how that influences sharks like across their entire group, so across all 9 orders. And so that's basically where my question comes in. I actually wanted to see if the proportions and shapes of these RU regions influence the lifestyles of sharks, and I want to do that by kind of using the neurocranium as a proxy for inferring the importance and patterns of their lifestyles. With that in mind, my study's goal was twofold. The first goal was to really examine whether there were any relationships between these RU proportions in sharks and their habitats, diets, and their body forms. And then my second goal was to see if there are any potential patterns between those features of the neurocranium with regard to their ecology and evolution. So to accomplish these goals, I examined and traced illustrations of the dorsal and ventral views of lots of skulls of different extant shark taxa from published literature that's listed right here. In addition, I obtained specimens from the following institutions listed here, and you can see they're abbreviated by code. I also snapped photos of these specimens and then recorded radiographic images of them. And I mentioned I already have published illustrations, but I want to include radiographic images so that if I have multiple samples from one species, I can average those out, get a better perspective of the entire group. And some specimens were only available in museum collections and could not be found online. And I'm also showing here that I'm also including published illustrations and some museum specimens as well of some fossil taxa, some fossil sharks. So the first thing I did once I got these data was to obtain measurements and then convert those into proportions of the anterior-posterior length of each region of RU, neurocranium. And here's how I define my limits. So the total cranial length, or TCL, is the combined lengths of the rostral length and the comparative cranial length. And the comparative cranial length is the length of the cranium from the distance of the anterior-most edge of the sharp nasal capsule down to the occipital centrum down here. And so I also define the rostral region as the distance from the anterior-most tip of the rostrum down to where the nasal capsule begins. And then if you look over here, the olfactory region's limits are determined by the beginning of that nasal capsule down to the posterior-most edge of the nasal capsule. The optic region limits are defined approximately by looking at the pre- and post-orbital processes and the extensions, which are basically anterolateral projections on the skull that kind of denote the range of the eye. And then the otic region is defined first by measuring a halfway point in a feature that's called the parietal fossa. It's kind of hard to see, but it's about right here. The parietal fossa is just a kind of small depression that varies in concavity and range depending on what you're looking at, but they are, and it's on the dorsal surface here of the skull, and it is something that is visible among all genera. And so once I found that halfway point and I went down to the occipital centrum, I know it says condyle, but it's supposed to be centrum, I then doubled that, and that's an approximate range of the semicircular canals, or in this case, the otic region for my study. Before we go on, I just want to mention that I did these measurements on the hammerhead sharks a little bit differently, because remember, I'm measuring the rostral region to capture the amount of electroreception that's happening. And because hammerhead sharks have a trend, they're transversely elongated, laterally elongated, rather than being longitudinal, I thought that their ampullary canals were more dispersed that way. And so what I did was I first measured from the lateralmost edge of the nasal capsule over to the midline of the neurocranium. That was like the halfway, and then what I did similar to the OT was then I multiplied by 2, so I get that full range of the approximate usage of the ampullary canals. You'll also see real quick that this line right here is something that you'll see on my illustrations, which is basically a line that denotes 2 different sides. This side, the left side, is going to be the dorsal side. This right side is going to be the ventral side. So that would be the same when you see those. So with respect to the linear measurements, I attempted to test and refute these 3 null hypotheses. The first one being that there is no significant difference among RU proportions and habitat types. The second one being that there is no significant difference between RU proportions and diet or feeding form. And then the third one being that there is no significant difference among RU proportions and body forms. The other major method I did was called geometric morphometric analysis. And this was a method that allowed me to look at shark skull shape more quantitatively. Now, to accomplish this, what I did was I did a principal component analysis, or PCA. And this is a method that allows me to analyze skull shape in a software. It's an effective method because, like I said, it quantitatively looks at skull shape and it eliminates maybe additional variables that, just additional data that wouldn't matter, such as size, position, and orientation when you're just trying to capture shape. Then I placed homologous anatomical points called homologous landmarks, which are these large dots on features of each region that kind of try to capture the shape of that. And then after that, I did small little dots called semi-landmarks in between that help capture the shape more quantitatively. So once I had all my tracings, I formatted and digitized them on software such as TPS Util 64 and TPS Dig 232. And then that's when I started plotting these different landmarks. And then once I had all of my skulls plotted like this, then I would run them in a software called Morpho-J, which basically allows me to process all of the skulls and all of the landmarks on those skulls, and then create scatter plots and group the different skull shapes based on similar features. You'll be kind of seeing that soon. I also want to point out that there are, oh, I'm sorry, you know what? Forgot to say that I ran this geometric morphometric analysis, and then I used some parameters to kind of look at shark ecology and evolution. One of them was an analysis that had scale bars, and one that didn't, to see if size and shape, they played a role. The other was by taxonomic order, and then the other was looking at it through the lens of habitat, through diet, through the two body forms, and then living status, so extant or extinct. Okay, now I do want to point out there are a few limitations to this. The first one is that I'm only looking at the genus level, so it's close to being specific, but it's not species level. So there might be some taxa that I'm missing from my analysis that might capture interspecific morphological variation, and so that would be excluded for that reason. The second one being that the otic region, which I talked to you about, is more of an approximation than an actual, full measurement, because like the other structures, you can actually see the areas and measure them, but this I had to kind of approximate, but it's the best that we could kind of come up with for the time. And then the third was that all of the analyses, it's limited in that it's two-dimensional dorsal ventral view, so it disregards potential other anatomical characteristics that might be present, as well as just the overall three-dimensionality of the diversity of shark skull shapes. And here are some of my tracings for all of the extant sharks. So this is all the shark species, or all the shark genera I looked at within every order except the Carcharrhiniformes, which is this order because it's the largest order. Yeah, so I have those. Oh, and just to note, I had 182 species that I did this for, and again, that has 9 orders, 37 families, and 102 genera. I mentioned I'm also looking at fossil sharks, and so I have 10 skulls of some fossil sharks there. So I have 10 species, that includes 5 orders, 8 families, and then 10 genera. Okay, so once I got my linear measurements, now I need to convert them into proportions. I have an example of how I did that for each of my skulls. So to find the proportion of the rostral length, or PRO, what I did was I divided the rostral region measurement by the total cranial length. And so then I got a proportion. And each of these proportions is between 0 and 1, so I get a good estimate of how they compare to one another. I did the other regions a little bit differently, where I divided each of them by the comparative cranial length instead. Now, I did this because the rostral length it varies a lot if you look at all the sharks. Some might even have a rostral length that exceeds their comparative cranial length. And so to make it more comparable, I wanted to just divide the rostral length by the TCL and the others by the CCL. What I did next was I put my data into 2 statistical tests, being the Kruskal-Wallis test and the Mann-Whitney test. I used the Kruskal-Wallis test to look at the relationship between my calculated linear proportions of the Rue regions and their habitat and diet types. And then I used the Mann-Whitney test to look at their relationship between linear proportions and body forms. So we'll talk about it more in a few slides, but these results show that there is a statistically significant relationship among nearly all proportions of Rue, apart from the optic proportion in the habitat forms and the optic proportion in the body forms. And here are the results in box plot form here. So you can kind of see the general trends that each major group has. Now for habitat types, you'll see that benthic sharks, they tend to have a smaller rostral proportion than the demersal and pelagic sharks. Pelagic sharks tend to have a smaller olfactory region compared to, say, the demersal and the benthic forms as well. You'll also see that benthic sharks have the smallest otic region among the others within this grouping. For diet types, durophagous sharks have the smallest rostral and optic regions, and they have the largest olfactory regions. And then for the body forms, you'll see that group A had a small rostral and otic regions, and they had larger olfactory regions, and it was inverse for group B. Okay, so for the geometric morphometrics, this plot kind of shows that there's one major clustering of sharks, and then there's the one major cluster, and then two kind of smaller clusters of distinct groups. PC1 and PC2, which are on the X and Y axis respectively, they cover 55% of the variation together. Just as a side note, PC1 and PC2 means principal component 1 and 2, and basically the first principal component captures the most variation, so it's going to show you what object or feature is the most variable among all the species. And then principal component 2, it shows you the second most variable, and so on and so forth. So the top left side of this plot indicates that sharks have a slender or a skinnier skull or neurocranium, and that they have a shorter rostrum, so these guys up here. And then if you look at the right side of the plot, this indicates that sharks have a long, longitudinally elongated rostrum and also a skinny neurocranium. And then if you look at the bottom cluster, these are practically the opposite of these, where you have laterally expanded optic regions as well as like a broad skull in general, and hence the widening of the skull for these lower values. So basically, to kind of sum that, there's two diverging trends, either an elongating rostrum or an expanding skull width. Now remember, I mentioned that there's PC1 and PC2. I did get PC3, 4, and 5, but the differences and the variation was much more negligible than the other two, and I thought the PC1 and 2 captured the most variation that was relevant for this study. Okay, so for my linear proportions, I think that the small olfactory, or rather the large olfactory region and the small rostral and otic regions in benthic sharks is likely because that olfactory region enhances their sense of smell when they're on the bottom and they're looking for prey with their sense of smell. And then it's commonly seen that sharks on the bottom occupy kind of a blunt snout with a acute sense of smell. And since they're not swimming actively above the ocean floor, or kind of actively swimming in general, I didn't think that vestibular balance was as important or necessary for them. So for demersal sharks, I think that they have a longer rostrum than the benthic ones because they are, in fact, swimming at least more than the benthic sharks, so they need to have at least some kind of hydrodynamicity as well as maybe some areas to help with electroreception. But also it's good to note that they have a large olfactory region relatively and a large otic region, so it's kind of like a step above from the benthic sharks, where they have good sense of smell because they're still above the ocean floor trying to find benthic food sources, but they're also swimming, so they have to have something to compensate for that. So pelagic sharks, I think, were aided by a longer rostrum because they're doing even more hydrodynamic swimming, arguably, as well as heightened electroreception, because they're often, it's useful when they're constantly moving around and it helps them detect food sources and things like that. And then the large otic region as well, because again, lots of different swimming and all types of wide spatial coverage. And then if you look at the feeding forms, the suspensivorous sharks, they tend to have a longer rostrum and then also large optic and otic regions. I put the asterisk up there because I only had 3 suspensivorous sharks that I could see. And this one here is the one that has the longest rostrum, which is the basking. But the other 2, which are the whale shark and the megamouth, they have kind of more blunt rostra. So I think that the basking here in the low sample size kind of makes that data look more like they have that a long rostrum. But I think that they have large optic regions because they are pelagic in lifestyle, so they do, potentially need to rely on visual cues and other things like that to find their food sources. And otic, again, for the same reason, they're pelagic. They undergo lots of wide spatial coverage, and even sharks like the megamouth will do DL vertical migration. Macrophagous taxa, I think that the reason I think they have these longer rostra and larger optic regions is because they tend to be reliant on electroreception to find their prey, as well as a large optic region because there's lots of predators in macrophagous forms, and they rely on good eyesight. And then kind of a smaller olfactory and otic region, because I would say that they would rely a little bit less on those than these senses here. And then the durophagous forms, those are aided by a larger olfactory region, and I think that helps them because durophagous sharks tend to be benthic and find benthic or bottom-dwelling food sources, and so that sense of smell helps them. And that small rostral region as well kind of creates like a blunt, kind of short, stubby snout for them, which is actually helpful for them to have a cranial skeleton in such a way that they can manipulate smaller objects in their mouth and perform suction. And then the smaller optic and otic region, again, because they're relying a little bit less on their sight and less on swimming, since they're kind of more up down on the substrate. For body forms, the group A sharks were found to be predominantly benthic because they have these large olfactory regions, and they were also found to have smaller rostral and otic regions. And so that made me think they must have similar kind of traits to these benthic forms. And then the pelagic forms kind of coincided with the group B sharks because they shared similar features, like a longer rostral region and longer otic regions for more active oceanic lifestyles. All right, so the major clustering of plots up here in this top left corner of the morphospace basically suggests that the vast majority of shark taxa kind of have one general skull shape or one general neurocranial morphology. And that kind of default is right here for central forest granulosus of having this slender skull and short rostrum. Now, most of the members of all of the orders kind of occupy this cluster here. There are some members of lamniformes and carcharrhiniformes that kind of go split either down or to the side. And this basically suggests that one lamniform, which is the goblin shark here, and then the saw sharks kind of diverge from this cluster by elongating their snouts longitudinally. And then if you look down at this cluster here, sharks like the hammerhead sharks kind of achieve snout elongation transversely or horizontally, as well as kind of broadening their skulls more so than these forms. And the whale shark is kind of like that to a little bit lesser extent. So I did not include skulls of batoids, which were skates and rays, which are close relatives of sharks. But I did include this fossil taxon, which is called Hybidus, which is a sister group to the sharks and rays. And I kind of used this as the basis of like the basal group for comparison, or the out group. And being its position here in the figure does kind of support the fact that sharks kind of started off with that more slender, short, robust neurocranial build. Now for habitat types, it kind of reveals overall that benthic taxa, if you look here at the purple outline there, they tend to have the kind of the most of the morphospace situated up here, so most of them, or they're all going to have a slender neurocranium, but the rostral length will vary in them. So some benthic sharks, like the angel sharks, are going to have very circular, blunt snouts, and then the saw sharks are going to have the longer snouts for the benthic forms. If you look at the demersal forms, which occupies the largest morphospace here, you can see that they have either very slender neurocrania, like this bird-beaked dogfish, and then if you go all the way down here, wide neurocrania, like the hammerhead shark. But they're both kind of situated toward the left end of this x-axis, which indicates that they don't have a more like a longer rostrum. They're longitudinally blunt. And then if you look at the pelagic forms, these are kind of situated in between the benthic and the demersal. And kind of if you see the extremes of the benthic forms, or rather the pelagic forms, so like the goblin shark here with that long longitudinal rostrum, or the whale shark here has that laterally or transversely expanded skull, you can see that these outliers kind of evolved their skulls a little bit differently. So in the pelagic taxa, you'll see there's some great variation with that. So for diet types, kind of want to just introduce that there are generalists and there are specialists. Generalists consume a wide variety of food sources. Specialists, they consume only one type of food source that they have evolved to be able to consume, and in most cases it's only that type of food source. So the observed plot distributions right here show that the macrophagous sharks, because they occupy this large morphospace, it shows that they're probably more of the generalist feeders, as opposed to more of the specialized feeders like the durophagous and this purple here, where again they eat those kind of heavy, hard-bodied foods, and they are more constrained in morphospace. And if you look at the suspensivorous sharks in the blue, I call this the suspensivory triangle. You can see that if you look, they're more wide, broad in their neurocranium than they are skinny, and they don't part from the megamouth, or part from the basking with a long rostrum. But this is also so because, again, we only have like a few taxa, but that wide skull allows them to kind of take in more water, and they can open their mouths and filter more water, as opposed to that smaller mouth of the durophagous sharks. And it's also worth noting that one extinct lamniform shark, Lamna milarke, was kind of in the plot. It's not included in this plot, but in subsequent plots, you'll see that its point is about right here, and this would support the idea that this was a suspensivorous filter feeder as well. All right, and as I mentioned, Stearns and Shimada 2020, they demonstrated that there were two basic body forms in sharks, the shallow body group A forms and the deep body group B forms. Remember, they noted that the majority of group A sharks were seen to be benthic, and the pelagic forms, or the group B sharks, were seen to be pelagic. This overall plot distribution of group A sharks in the orange there is nearly identical if you look at the benthic plot. And then you could argue as well that the plot of the group B sharks is very similar if you were to combine the demersal and pelagic plot boundaries. All right. This is the plot of the living status, so extant versus extinct sharks. You can see that the extant covers more morphospace, but I would say that the fossil forms covering that more limited morphospaces, probably more due to the lack of available fossil specimens than that it's just that they're that kind of limited in their neurocranial diversity. And so another thing that I want to highlight, though, is that these red dots are lamniform, the order Lamniformes. And if you see that, it kind of shows that the lamniform sharks, if you include Achilolamna, like I said, as well as some others like Heinrichia almonensis, that would basically show that lamniform sharks achieve the greatest diversity in skull shape, if we are to go with that. So my study is significant because these results provide important information for conservation biology with respect to sensory biology. As an example, it's conceivable from my study that the benthic sharks are particularly prone to extinction because of these rising sea temperatures that actually create more acidic conditions in their habitats, and higher acidic conditions actually decreases the effectiveness of their olfactory capabilities, so they aren't able to get food sources as easily. Other examples would be for the macrophagous sharks. These warming oceans effectively kind of decrease the mineralization in their cartilaginous endoskeletons. And what that does is it makes them more dense, and so they have to work harder to stay up and afloat. And since they're compromising that, makes them more susceptible to being, fished or hunted by their natural predators. Because of these regions, I think that the sensory biology of sharks is very important, especially for their future conservation efforts. So what I want us to take away from this is that we know that sharks occupy a wide range of ecological niches, and that the neurocranium is a suitable tool for looking at their sensory regions. There is a form-function relationship between shark lifestyles, body forms, and most of the regions. There was one basic skull shape in sharks throughout their evolutionary history, and that only diverges in two extreme ways. And it's also interesting to note here that sharks occupy kind of this area of the morphospace, a little bit here, mostly here, a little bit there, but these areas that are empty, I'm wondering if those are because we have a lack of specimens, or is it really because sharks are unable to achieve a kind of a skull form that would be in those areas? And then lastly, that the shark neurocranium offers insight for conservation biology through the lens of sensory biology. Acknowledgements. Really want to thank Dr. Shimada for all of his help. Wouldn't be able to do any of this without him. Dr. Aguirre and Dr. Nordstrom, committee members, and Dr. Brooke as well for her help throughout my academic development, as well as lots of people in the Shimada lab and all these other individuals. Let's see, my references. And yeah, thank you so much for being here. I really appreciate this, and yeah, I'm happy to take any questions.
Nordstrom and I will have a lot of questions later. After this, we'll have a